Welcome to the UNH Judd Gregg Marine Research Complex, located 15 miles from the Durham campus at the mouth of Portsmouth Harbor in Newcastle, New Hampshire. This complex supports research, education, and outreach in all aspects of marine biology, oceanography, and ocean engineering, with an emphasis on marine biology and ecology, aquaculture, acoustics and ocean mapping, invasive species, autonomous surface vehicle research, ocean acidification, and renewable energy. The two main facilities that make up the marine complex are the Coastal Marine Laboratory and the Marine Research Pier. Here you see the Coastal Marine Lab, which is a year-round marine laboratory facility with wet and dry lab infrastructure. The wet lab contains tanks that range from 3 liters to 1,600 liters, with full-strength seawater capabilities, ultraviolet filtration systems, and a variety of supporting infrastructure, including both flow-through and recirculating marine systems. Numerous cold-water marine species have been studied and cultured at the Coastal Marine Laboratory, including finfish, shellfish, crustaceans, echinoderms, gastropods, and algae. Here you can see some lumpfish that are currently being grown at the lab by Research Associate Professor Elizabeth Fairchild to investigate their use as cleaner fish in cage-reared salmonid farms in New England. In the dry lab, there are several workstations, compound and dissecting microscopes with supporting camera and imaging software, balances, desiccators, and other lab supplies. Down at the water's edge is the Marine Research Pier. It is 320 feet long with fixed and floating docks, which is home to UNH's fleet of vessels, including the RV Gulf Challenger and the RV Gulf Surveyor. The RV Gulf Challenger is a 50-foot U.S. Coast Guard inspected vehicle capable of carrying 39 passengers on day trips and 6 scientists on overnight cruises. The RV Gulf Surveyor is a 48-foot, twin-screw, geared diesel propeller-driven catamaran with a state-of-the-art platform for hydrographic research. The Marine School also maintains a fleet of other smaller boats at the pier that are available for research use. The pier is outfitted with a 3-ton capacity crane, and experimental enclosures are located under the fixed pier. Shore support is also provided at the pier for New Hampshire Sea Grant's integrated multi-trophic aquaculture research that takes place on UNH's Aquafort, floating just offshore of the Marine Research Complex. Funded by the Sea Grant National Aquaculture Initiative's 2018 Aquaculture Research Award, the Aquafort was constructed, deployed, and tested by a team at New Hampshire Sea Grant and UNH alongside local fishermen. At the Aquafort, researchers grow sugar kelp, blue mussels, steelhead trout, and other species in a self-contained aquaculture system that allows multiple species to grow at the same time within and around a single floating structure. Net pens of steelhead trout are surrounded by blue mussels and sugar kelp, which absorb nitrogen waste released by the trout. This nitrogen-neutral system has the potential to bring aquaculture to communities around the world, looking to grow local seafood and support working waterfronts in an environmentally and economically sustainable way. Thank you for touring the University of New Hampshire's Judd Gregg Marine Research Complex.
Hi, my name is Elizabeth Harvey, and I run the UNH Phytolab, short for phytoplankton. And we study all things microbial biology in the ocean. So particularly some things that we're interested in is water quality. So we can enumerate anything from larger zooplankton down to viruses. We've had a particular experience working with harmful algae and looking at harmful algal blooms. And then we have an interest in human health in the ocean. So understanding pathogens and particularly how potential marine microbes can be vectors of some human pathogens. We use a variety of methodology. So in the lab, we have a flow cam, which takes images of phytoplankton in the water. So we can get some idea of who's there, how many of them are there in terms of species composition and community composition. The really interesting thing is that FlowCam has been used for a lot of different purposes, analyzing microplastics in the water, looking at silica particles or sand particles in the water. A lot of wastewater treatment plants are using a FlowCam to look at how clean or effective their wastewater treatment plant or procedures are working. So there's a wide variety of uses of the FlowCam, all of which are particularly interesting. We also have a flow cytometer, which looks at the very small size classes of phytoplankton, so anything between about 2 and 5 microns in size. So we can count bacteria and viruses on here as well as phytoplankton. We can also use a variety of diagnostic stains to identify particular species if we would like, or some physiological process that they're undergoing. We also have used eDNA to look at changes over time of different marine populations in the water column. And then we also can do pathogenic bacteria enumeration. Here are some examples of results that we have generated from our research. So here's a graph of chlorophyll concentration, both along the outer coast of New Hampshire, but also in Great Bay. So you can see the relative sort of peaks and valleys of chlorophyll. And then using the FlowCam, we can also identify what species are there. So these peaks here are attributed to these species names you see here. These are all diatoms that aren't toxic, but it's really interesting to know in terms of, you know, not all phytoplankton is made the same. So if you're thinking about aquaculture or something like that, you know, the type of species of phytoplankton that are present in the water column make a difference in terms of nutritional content for things that are larger growing in that same water. And also we do look out for harmful algal blooms as well. We can see Studnitia and Alexandrium pretty routinely. We also, here's an example from work that we've done in coastal Georgia, where we've looked at relative abundance of different organisms. In this case, I'm showing the relative abundance of 2 oyster species. So we can use eDNA to track relative population abundance over time. And we can do this for pretty much any species that we're interested in, you know, anywhere from, you know, oysters all the way down to pathogenic bacteria. Please don't hesitate to reach out. I'm really interested. I think particularly our lab could have really nice synergies with people interested in aquaculture applications, anything to do with water quality, and I look forward to hearing from you soon. Thank you. Bye.
My name is Greg Moore, and I'm an associate professor in the Department of Biological Sciences. I'm a restoration ecologist working in temperate and tropical wetlands, particularly along the coast, working in systems like salt marshes, dunes, and mangrove habitats. Our coastal habitat restoration team, together with colleagues from UNH, other universities and NGOs, as well as coastal communities from New England to the wider Caribbean, focus on protecting, enhancing, and restoring wetland habitats. Building on our ongoing work in the region, we recently received a significant NIFWIF grant to address marsh and dune resilience across 2 New England states, New Hampshire and Massachusetts. We're focused primarily on the Great Marsh, which includes the Hampton Seabrook estuary. We're also working to inform coastal communities of flood risks in the Hampton Seabrook estuary through innovative sensor placement and developing better web access and data interface tools. We're also exploring new ways to help marshes keep pace with sea level rise through innovations in thin layer sediment placement and experimentation. Meanwhile, we have projects and initiatives working in post-hurricane restoration of mangrove habitats throughout the Caribbean, most recently in the British Virgin Islands. Links to industry for our programs may include innovations to improve the technology of data collection and tech transfer, as well as how data are shared and accessed by the public, such as developing better user interfaces. We're also interested in scaling up production of native plantings for niche markets. We're interested in finding new and exciting ways to collaborate. Thank you.
Hi, I'm Allison Watts, and I want to talk to you a little bit about some really cool work we have been doing using eDNA to characterize marine systems. So you've probably heard about eDNA. It's kind of a shorthand form of the word environmental DNA. And environmental DNA is just DNA that's present in the environment, whether it's water, whether it's sediment. All organisms pretty much shed DNA through their scales, their skin. If they go to the bathroom, if they reproduce, they're releasing DNA into the environment. And we can collect samples of the water or the sediment. We extract the DNA, and we use that to see what organisms are present. We've been using this really successfully for a bunch of different applications. We've been doing a bunch of work detecting fish in estuarine systems, and we find that using the water samples, we are able to detect pretty much all the fish that we expect to see present in that system. We can also look for different types of organisms. So for instance, this is some samples that we took from deep sites in the Atlantic Ocean. And for each liter of water that we pulled up, we detected generally a couple of hundred different species. And most of those are microorganisms. So there might be fungi or diatoms or bacteria, but we also see fragments from larger organisms like fish or whales. We didn't actually see whales in any of these samples, but people do, and that is a way of detecting larger mammals. We can also use that whole community to look at change. So these are samples again from Great Bay, where we sampled in June, August, and October. And over time, we see that the biological community shifts, which you would expect over the course of the summer. But this is a way in which we can actually quantify how that changes and which species become dominant at which times of year. We can also use this method to see how ecosystems shift in response to either restoration or to a stressor. So for instance, if you are interested in wind farms and how those wind farms might affect the biology of a location, we can use eDNA to characterize the biology that's present before the installation occurs, during the installation, and then continuously monitoring over time as the installation matures. And this is being done in several different locations, and we're finding that it works extremely well as a complement to other types of monitoring methods. So you've probably heard of eDNA. You're also probably familiar with a lot of different monitoring methods. And what I'd really like you to get out of this is you should really be considering eDNA either as a standalone or as an add-on to existing monitoring methods. It will make a difference to the types of biology you are able to detect, really. And I'm happy to talk to you about it if you want to hear more about it. Thank you very much for stopping by.
Hi everyone, my name is Dr. Easton White. I'm an assistant professor in the biology department here at the University of New Hampshire. I run the quantitative marine ecology lab at the university. Of course, the lab is not just me, it's made of a bunch of different people, including graduate students, undergrads, researchers. There's just a handful of people that are in the lab currently, all working on different projects that I'll talk a little bit about today. So what is the lab? What do we do? So the members of the lab are committed to producing and sharing high quality scientific research in order to both advance our understanding of natural systems, but also to inform their management. So we think a lot about applied aspects as well. We also committed to thinking about ideas of diversity, inclusion, and equity in our work. And that's through teaching, outreach, and research as well. The lab focuses on a few different core components, and then we have people doing a lot of different projects here, but we have kind of the intersection in marine systems of extreme events, improving ecological or environmental management, and then socio-ecological systems. So what happens when you include humans and ecosystems together in the same system? We use a lot of different tools, long-term data sets, mathematical models, statistical models, and fieldwork. So we use all of this, in particular this quantitative side, in order to answer a lot of questions around these areas. Research background of the lab is really a mix between developing mathematical models and being able to predict things, and also doing statistics, data science, big data, various different approaches depending on what the project requires. A couple current research projects, and you'll see kind of the breadth of research that we have in the lab. We think a lot about harmful algal blooms and hurricanes and how they affect natural resource use. We've done a lot of work so far on COVID-19 and how that is affecting ocean-related industries like shipping, seafood, other industries. A lot of this work is using big data, particularly tracking vessel traffic using satellite technology. We also think a lot about how to improve environmental monitoring. So whether that's doing things like eDNA or acoustic telemetry or other types of environmental monitoring, and how to make that more cost effective for whoever is implementing that technology. And last, we also use and think a lot about developing new novel data sources. So things like Twitter or Google Analytics as indicators of things that you might be interested in marine systems. Our work has also been highlighted in a lot of different places. So this is just one particular example. This is one paper that we published last year. So this was covered in 200 news outlets and cited in policy documents like the U.S. Congressional Research Service, for example. And it just really, I think, really shows that we're not only interested in doing, basic science, but also really applied work that is a lot of interest to people around the world. And I look forward to chatting with you over the next couple of days.
Hello, my name is Kaizia Vogel, and I'm a research associate professor at the University of New Hampshire with the Ocean Process Analysis Laboratory, OPAL, and we are one of the 5 centers in the Institute for the Study of Earth, Oceans, and Space. And I happen to be the director of the center. Now, OPAL is comprised of 6 different research groups. My group is the microbial biogeochemistry lab down here, but we also have PIs that study coastal carbon, coastal circulation modeling, ocean nutrient biogeochemistry, ocean observing, and remote sensing. So our group is really focused on biogeochemical processes and physical processes that we study in the ocean, not only in the Gulf of Maine, but in other marine environments as well. And we are mainly interested and mainly enthusiastic about this process that we call the biological pump. And on this slide here, the schematic, you see the major factors that affect the biological pump. And it looks a little bit confusing at first, maybe, if you haven't seen this before. But what this really boils down to are the processes that affect the flux of atmospheric CO2, as you can see here, into the ocean, and then the processing of this CO2 by marine organisms, which then eventually sequester this atmospheric CO2 that they fix in the deep ocean, and which again gets transported down to the deep ocean and then eventually buried into the seafloor, which is really an important aspect of the ocean uptake of atmospheric CO2 and storage and regulating the global CO2 fluxes on this planet. And my group is mainly interested in these, and I highlight these aggregates here in the middle of this schematic, the marine aggregates. We call them marine snow, actually. So these particles, they form from phytoplankton production, zooplankton grazing, and fecal pellets, and bacterial interactions with the organic matter. And these particles are millimeter sized. You can see here on the right, those are microscopic images of marine aggregates or marine snow, and they can sink pretty fast through the water column, 100 meters per day. And what I'm also really interested in are these, this picture B and C here, how these marine aggregates interact with marine contaminants, actually. So we've done work on the effects of spilled oil on marine snow or marine aggregate formation. You can see here in picture B, those black dots is actually crude oil. And we've done experiments and field observations in the Gulf of Mexico following the BP oil spill. And we found out that the sinking of marine snow with oil, we call them marine oil snow, really was a substantial sink of the spilled oil in the Gulf of Mexico. I've also recently started working with microplastics and plastic contamination, and we also found that these particles are important drivers of plastic contaminants in the water column. So I look forward to fruitful discussions about my research in the microbial biology chemistry lab, but in OPAL in general. Thank you very much.
Hello, I'm Michael Pallas, and I'm here to talk a little bit about our group's capabilities as part of the UNH Blue Economy Sandpit. I'm an associate professor in the Department of Earth Sciences, and I run a geospatial and remote sensing lab. Our group's study focuses on imagery of the Earth to look at like forest structure, snow characterization, and cyanobacteria and water quality. Our work began using remote sensing techniques to look at the impact of tropical logging in Brazil. From there, we started to look at LiDAR remote sensing to look at forest structure. We've also developed algorithms that delineate individual trees and crowns and counts them, such as this example. And we've also deployed a terrestrial LiDAR system at various places around the planet. Our lab in Morse Hall has thermal LiDAR hyperspectral sensors with various drones that can be deployed. We've been looking at hyperspectral data from Lake Sandpit to look at cyanobacteria, and hyperspectral data to look at foliar nitrogen at Harvard Forest. We've also been flying snow depth estimates using LiDAR at Thompson Farm here in Durham. Here's an example of kind of the LiDAR system calculating where the trees are and penetrating to look at the baseline ground. We've also done work with remote sensing using a fixed wing drone in Eastern Oregon, and also used the same fixed wing drone in Sweden above the Arctic Circle to look at permafrost collapse. And here we have a near infrared camera on our drone. We've flown this site over 8 years. We skipped 2020 due to COVID and have a great time series of the permafrost collapse. We've also expanded and done hyperspectral work in Northern Canada. And here we're comparing different vegetation cover types and trying to predict them using neural networks. We've also done some geospatial modeling of past human impacts on the Amazon, the whole Amazon basin. We've also been using Corona satellite imagery, spy satellite imagery from the 1960s to look at fire burn scars in the Amazon. We're currently looking at fires in the Amazon from 2012 to 2022 with ongoing work. Thanks so much for your time, and I'd be happy to answer any questions about our drone capabilities and our remote sensing capabilities. Thanks.
Hello, my name is Nathan Logshag. I'm an assistant professor of ocean engineering in the Department of Mechanical Engineering here at UNH. I'm going to give you a brief overview of my research. The ocean is a vast, vibrant, dynamic environment. A crucial component of our understanding its dynamics lies in describing the interaction of the ocean with the atmosphere. Now, these interactions have large-scale consequences, but they're generally mediated by processes on the scale of individual surface waves. This is well below the resolution of our weather and climate models. An additional complication is that our observational techniques tend to give either a 1D time series, say from a fixed sensor, or a snapshot, say from a satellite. This limits our understanding of the relevant physical phenomena and constrains our ability to validate numerical models. As an example, let's look at a wave buoy. Wave buoys record and telemeter surface wave kinematics at a single point. They can sit out in the ocean for years, giving us long time series of wave conditions at their location. This is a 20-minute segment of the water surface vertical displacement data observed by a buoy. What these buoys do not give us, however, is a direct view of the full wave field around them. We have ways of interpreting the data recorded at one point to infer directional characteristics, but there are substantial limitations to these approaches. At this point, I'm going to tell you what I personally worked on in the past. 2 years ago, I was part of a field observational campaign with Dr. Chris Zappa at Columbia University. We outfitted the Air Sea Interaction Tower south of Martha's Vineyard with a number of instruments, among them a polarimetric camera. The polarimetric camera allows us to observe the spatiotemporal evolution of the sea surface topography within its optical field of view. Here's a representative example of the surface wave information we can access using this instrument. Millimeter scale resolution in length and height over a meter scale footprint. Now, that field of view is too small for us to describe the larger surface gravity waves, which are directly relevant for coastal and ocean engineering applications. For this reason, I'm looking to deploy the sensing capability on a stable, remotely piloted airborne platform. A multi-rotor unoccupied aerial system would give us synoptic measurement capability of the surface wave field development in space and time. This really expands the scope of research that can be performed by our group and opens the door to future collaborations. Now with that, I'd like to thank you for your time. If you'd like to learn more about our research group, what we do, this QR code will take you to our website. Have a great day.
Hi, everyone. My name is Nathan Fury. I'm an assistant professor here in the Department of Biological Sciences, and I am the head of the Fish and Movement Ecology Lab here at UNH. I am a fish biologist, but I'm also specialized in marine animal movements. And so from a practical sense, this means that my lab takes a lot of time in trying to define the space use, habitats used or selected for by animals, how quickly do they move or migrate. In some instances, we can estimate survival rates, and we can incorporate a lot of this information to try to develop habitat distribution models to predict where and when animals will be located. One of the main methods that my lab uses to investigate movement ecology in marine environments is called acoustic telemetry. So we can take an animal such as a striped bass, put an electronic tag in it that's going to emit a unique acoustic signal. And that unique acoustic signal can be heard by hydrophones or listening stations placed in both saltwater, estuarine, as well as freshwater systems. It gives us individual level information on their movements. And my lab has tagged thousands of animals across ecosystems and taxa, from very small fish all the way up to large pelagic animals and even crustaceans. And we're also now working with others that use other technologies such as GPS tags on seabirds and applying some of our quantitative tools to these critters as well. And so these types of studies can be done really across spatial and temporal scales. So whether it's tracking a salmon migration for over 1,000 kilometers, or whether it's a baywise study such as the movements of rainbow smelt in the top left, or we can even use positioning systems where we can, to just a couple square meters, look at the movements of an individual animal relative to an anthropogenic feature such as being showed in the bottom right-hand side. However, it's not just looking at where do these animals go, but also trying to understand the ecology behind their behavior. So here we've applied a statistical model known as a hidden Markov model to the movements of these GPS tagged seabirds. And those blue dots that you're seeing shown up are areas that are likely to be foraging grounds for these terns. And this idea of foraging is really important in our lab. So we also use stomach content studies to look at what animals are feeding on and how quickly are they feeding. And then what is also the quality of that diet. So as that diet changes from one season to another, or as waters warm, what does that actually mean for their growth potential, which is going to influence their fecundity or their population productivity as well. And so whether it's movements or feeding, we try to integrate those as much as possible to understand the impacts of anthropogenic effects, whether it's increasing temperatures, wind development, aquaculture, or kind of more traditional blue economy measures such as commercial fishing. So with that, thank you so much. My email is at the bottom.
Hello, my name is Michael Chambers. I'm an aquaculture specialist at the School of Marine Science and Ocean Engineering. Today, I'm going to talk about marine aquaculture at UNH. Several different forms of aquaculture. There's land-based recirculating aquaculture that uses biological filtration. We have flow-through aquaculture systems that use seawater, pump it through and put it back in the water. We have coastal near-shore cage culture, suspended aquaculture, as well as open ocean aquaculture. The main facilities at UNH that aid this type of research include the Jared Chase Ocean Engineering Lab in Durham, New Hampshire, the Jackson Estuarine Laboratory in Great Bay, the Judd Gregg Marine Research Complex in Newcastle, where we have vessels and pier services. We have a nearshore IMTA farm, the Shoals Marine Laboratory in Appledore, and the offshore technology park just south of there. Oyster farming is the biggest aquaculture business in New Hampshire. There's over 15 businesses now that produce about $500,000 annually in sales of selling oysters to restaurants and seafood vendors. There are also ecosystem benefits of this species by filtering the nutrients out of the water. Nearshore, we've been developing small-scale farming systems to train fishermen. The system has steelhead trout in the center, and then we have kelp and mussel droppers suspended around them that take up the nutrients from the fish. This allows the farmer to sell 3 different seafood products, but it also helps clean up the environment. Here we are training a number of fishermen from the local area on this technology. We hope they can do this either part-time or possibly full-time in the future. We're also developing submerged kelp aquaculture lines. This is 10 feet below surface, so vessels can go over this. This grows in about 6 months' time and then goes to the marketplace. This type of technology has allowed us to secure a DOE funding to develop large-scale macroalgae farming systems that can be used for biofuel. But one of our concerns is marine mammals, and so we're looking at whale-friendly gear that won't allow them to entangle in this type of system. We also have offshore submerged longline aquaculture of mussels. And finally, I'll end up with the offshore technology park. This is located 9 miles offshore and about 185 feet of water. We've tested numerous types of aquaculture systems, air feeding systems, telemetry, mostly submerged systems, and we've grown a number of species of fish there. We really see this as a business model, as an incubator for new technologies to come in and be tested at the site. Thank you.